Hello and welcome to this week's What Were They Thinking? Among the news items we have for you this week are the unusually high percentage of side effects that aren't actual side effects, the benefits for both men and women when it comes to being vaccinated from COVID-19 and avoiding some of the very undesirable side effects, the bizarre information surrounding not only last week's pig-to-human heart transplant, but other research that's using pig kidneys to transplant these to humans instead. There are also two interesting astronomical news items. One is about the impact of space on astronauts' red blood cells, and the other is the Perseverance rover, and that Perseverance has encountered a molehill and made a mountain out of it. Let's start with the topical news about vaccines for the CCP virus and side effects to vaccines. Notably, how one relatively large-scale study has found that more than 3/4 of the side effects experienced by people may be effectively a placebo effect, something known as a nocebo. Nocebo as in unpleasant placebo. This has a big implication for the reticence for people to take up the vaccine. Many argue that they don't want to be vaccinated because of the period of great discomfort and unhappiness for several days after the vaccine's administered. If in fact the vast majority of these symptoms aren't real, but instead initiated by the mind, that may change their outlook on things. The way this has been figured out is that it was a meta-analysis. They took large numbers of high-quality randomized trials, 12 of them, and they had their placebo groups and their not placebo groups. 76% of the adverse reactions, things like headache, fever, and chills, were there for the group that had the vaccine, and 52% for those who had the vaccine for the second dose. The catch to this group, they didn't get the vaccine, they had a placebo. There was one noteworthy statistic though. That is the local side effects to the site of vaccination. 16% of placebo recipients identified issues with the site of injection, swelling, pain, and similar. 67% of the vaccine group reported those sorts of symptoms. The sort of symptoms you'd expect to see when you have localized inflammation from the immune system being active. This would indicate that there is a significant difference in the local symptoms, but not systemic symptoms between the two groups. One of the biggest groups that are reticent to be vaccinated are pregnant women, and understandably so. However, there's no evidence to say that they should not be vaccinated, especially towards the latter end of pregnancy. That's where a new study comes in that looked at 150,000 or so pregnancies. These pregnancies were in 130,000 women and from March 2020 to October 2021, so about 18 months. In this 130,000 strong woman cohort and 145,000 strong pregnancies, there were 450 premature births. This is either when it happens in the womb or during the newborn period. All were in unvaccinated women. There was a rate of death within 28 days of COVID-19 diagnoses, and that there was a significantly higher risk in those who were unvaccinated. There was a nearly 80% incidence of infection in unvaccinated women, 91% for hospitalization, and 98% for critical admissions. That shows all over there is huge benefit to getting the vaccine if you're pregnant or looking to become pregnant. If nothing else, it drastically improves the odds of successfully carrying a child to birth and them surviving the newborn period. A phrase that hasn't really been needed since people had to worry whether or not the milkman was going to have his horse and wagon working that morning. Another common argument with the CCP virus is that the vaccines cause fertility issues. As we've demonstrated in past weeks already, no, neither men nor women have fertility issues caused by the vaccine. Infection with the CCP virus, on the other hand, can cause fertility issues and other complications. For example, the case study of a man who had agonizing penis pain due to a side effect of infection with the CCP virus. The study comes out of Iran, and it describes how a blood clot developed in the man's penis, specifically in the shaft of the penis. This is the area that would become engorged and filled with blood when aroused. In fact, this appears to have been exactly how it occurred. Following an erection while having sex, the 41-year-old man experienced pain and was referred to a urologist. Upon scanning his penis, they found that he had a blood clot deep in the vein of the penis, and that this was what was causing the blood not to be able to get to the end of the penis, and this was obviously cutting off oxygen supply. When this happens, parts of it will begin to die and send out chemo signals that say, "This hurts, fix it." The article itself provides a small sampling of other case studies, let's say, of similarly natured events in other men who had complications with their smaller tools. Going from COVID news to other medical news, and the interesting article from last week, which was based on the idea of taking a genetically modified pig and transplanting its heart into a human. This was made possible because of those genetic modifications. The problem with that is that the entire technology and method has been made proprietary, and so knowing what's happening isn't anywhere near as easy to do. After all, the company wants to be able to make a profit out of their research. This is where other, less secretive research comes into play. This week, we're looking at the ability to transplant a pig kidney into a human. Admittedly a brain-dead human, but the issues are the same. It's proof of concept before moving on to trying to get approval to try it in actual living humans. Unfortunately, due to various circumstances, it took 5 days to get the transplant up and running, and it lasted for 3 days, before severe hemorrhage drained the body of all of the blood and led to what could arguably be described as a premature termination. The 3 days for which this happened are quite interesting for what they showed. The first was unexpected problems with one of the donor kidneys. It appears that it didn't seem to be working properly, and throughout the entirety of the 72 hours the experiment ran, there wasn't any real working capacity to it. The other kidney, on the other hand, started producing urine almost immediately, and as such, it was able to be considered at least largely successful. Unfortunately, by the end of the 3rd day, the functioning kidney had demonstrated damage to the tubules within it, which appears to have been caused prospectively by some sort of immunological pathway that the researchers thought they had mitigated, but may not have been able to do so if an alternative pathway was still effective. Going from extremely cutting-edge technology to archaeological news from millions of years ago. In fact, arguably up to about 150 million years ago. It's the discovery of a fossilized flower. But it's not just a flower for the sake of finding fossils, but rather the missing link in trying to understand some of the complexities and, frankly, unknowns of the development of flowers. We know what some of the earliest flowers were like, and how they developed over time to a certain point. But these weren't flowers as we know them today. Instead, they were in a different category. Gymnosperm. They're distinct and separate until we get to what we think of as a flower today, with leaf buds, petals, and so on. All of these are needed for it to be able to sexually reproduce. How do you go from an enclosed thing, almost like a pine cone, to something complex like a flower? There's a big gap between them, and that's where this fossil fits in. It helps to figure out just how plants leapt at a anatomical and physiological level between these two states. It matches with the genetic data that we currently have available, which is the important part there. It's a physical representation and demonstration of what we can see in genetics. Going from the missing link in plants to research in general, but research in general when it comes to genomics. A big project is underway to try and sequence every major complex species on Earth. That is about just under 2 million species in total. The plan is to try and get these roughly 2 million species all sequenced in about 10 years. That is a really big ask. The Human Genome Project took 13 years to complete, and that's one species done imperfectly. They want to achieve 2 million species in 10 years. That is ambitious. Very ambitious. It's true that research like this that's going to cover so many different species could be very important in some research areas. It's also incredibly useful if we're just trying to keep a bank of everything we know about the world. Some species like mice, fruit flies, and yeast, they are all extremely well understood. Most of them can have their genome completely described. Other species, like say, bears, salmon, and other less complex, let's say, creatures, aren't anywhere near as well understood. If we're trying to see differences in genes and activities and all sorts of other details, having so many different species, especially species that have almost nothing in common with humans, like say monotremes like the platypus or echidna, that could be really helpful. Not for reasons that are necessarily obvious now, but in answering questions that we don't yet know. There's also application for this in terms of evolutionary biology. Much like with flowers and the ability to trace the commonality of different genes throughout history, something similar could be seen with the kinds of genes that are present, missing, or otherwise weirdly altered in different species across everything that they're looking at. The next piece of news is also related to history and genetics to an extent, but more specifically, it regards the formation of proteins and how they may have evolved over time. Much like with the previous article where you could draw comparisons between different species and where they split off and how they diverged to create new species again, you can take a look at the different ways proteins can form to achieve certain results. That's what this article did. They were looking at the idea that there are certain proteins that function to be able to interact with metals. The very earliest organisms on Earth would need some way to be able to deal with oxygen, and having metals on hand is perhaps one of the best ways to do this. It allows for the transfers of electrons and more. In order for that to happen, and looking at it as a primordial soup environment, they took different computational models and tried to figure out how they could make a protein that would be able to functionally bind the metal and then be able to function within an organism. They found that there's actually a surprising number of similar ways to do this, and that different proteins can demonstrate these repeatedly, not just within the metal binding region, but in other regions. This means it could be a highly conserved sequence in the protein, and by doing that, these same things can show up again and again, but be used different ways. They describe it as being somewhat like a Lego brick, relatively uniform, but when put in conjunction with others, can create very different structures from the same beginning parts. This sort of research is not just important if we're looking beyond our own planet for life, whether that's life that's already existed or life that's coming into being. It has a role to play in research that we have now and problems we face now, specifically with astronauts in space. Going into space is not easy on an astronaut's body. Some of the more readily recognized symptoms of being an astronaut are stunted height. You lose some height due to bone weakening and then compression when you return to Earth. There are also other issues attached to it, but one of the weirder phenomena is called space anemia. Basically, it leads to a short-term issue where the body has red blood cells undergo damage and be destroyed. This leads to destruction of red blood cells by the immune system and other mechanisms, and so there's not enough red blood cells circulating around the body. There can be in excess of a 50% increase in turnover in red blood cells, but not an increase in production. That's just over 6 months. What gets even weirder is that after 3 to 4 months of being back on Earth, red blood cell levels had not returned to normal because the body continued to destroy 30% more red blood cells than they did before going into space. This is the kind of thing that could be very important when we're talking about either colonizing other planets or extending the duration of astronauts' stay in space. The final news item we have for you this week regards Perseverance and the fact that it's unable to surmount the problems posed by pebbles at the moment. Something that a child may kick down the street or along a path, throw into a river, and so on. These are the things that are proving too much for now for Perseverance. The small rocks managed to get into a part of the carousel, and unfortunately the carousel must move. If it doesn't move, it can't do its job. If they were to be too aggressive and do something wrong, they might become wedged permanently, thereby making it impossible for Perseverance to do anything else. This is why NASA is taking its very cautious time trying to figure out just how they're going to get rid of these rocks. That's all the news we have for you this week. Thank you for watching. If you found it interesting, consider liking, sharing, and subscribing. Please post any comments, questions, or suggestions that you have below.